as you might guess from this and from what David has said, I run a lab at the Eunice Kennedy Shriver National Institute of Child Health and Human Development, where we watch monkeys grow up. Specifically rhesus monkeys, a remarkable species, very much like us. They share 95% of the same genes that we do. They are a success story. They're the second most successful species of primates in the world. We're the first. They're the second. There are more rhesus monkeys living in the wild in a larger geographic range, during a larger range of climates than any other species of non-human primates. They come from the Indian subcontinent. You can find them everywhere from the middle of India's largest cities to the remote areas in Tibet and Afghanistan and Pakistan. They can live in rainforests, they can live on the edges of deserts, and they can live up to 14,000 feet in the Himalayas, so they are extraordinarily adaptive, and in that way they're very much like us, unlike most other species of primates. My lab has focused since its inception on the study of individual differences in personality or temperament of the monkeys that we watch grow up, and we know, for example, that about 20% of the monkeys in the colony that we maintain at my lab and that we have been able to see in the wild, about 20% are unusually fearful and anxious. They're prone to anxiety or depressive-like disorders. There's another 5 to 10% of the population who, from early on, have problems with control. They're impulsive, they do stupid things, they get into trouble, and they're unusually aggressive. And we now know that these characteristics show up very early in life. In the absence of major environmental change, they are stable. These characteristics are stable from infancy through the childhood years, through adolescence, and even into adulthood. But we also know, and these characteristics tend to be highly heritable, so genes are clearly involved. But we also know that early experiences, as David has alluded to, are crucial as well in shaping these patterns, and that the effect of early experiences show up not only in their behavioral repertories and their emotional regulation, but also in their neuroendocrine function, how they handle their stress hormones, their neurotransmitter characteristics, their brain structure and function, and we now know even in the way their genes are expressed. So my lab is, as David suggested, not on the main NIH campus in Bethesda, but it's in the countryside near a little town called Poolesville, about 40 miles up the river from downtown DC. Here's the Potomac River. This is the Maryland side, nice and green. This is the Virginia side. Suburban sprawl, that shows you what good zoning laws can do for you. We could never have our lab in Northern Virginia, but here it's an agricultural zone. The yellow boundaries, the yellow dotted lines are the grounds of the NIH Animal Center, about a square mile, a mile on each side. And in red is my laboratory, where we measure our lab space not in terms of square feet, as on the main campus, but in terms of acres. We're very fortunate in that respect. And in a place like this, or in the wild, these monkeys live in large social groups called troops. In the wild, the troops can range in size between 30 or 40 individuals on the small end, or up to several hundred on the large end. But whether the troops are large or small, they all have the same basic social structure. Every troop is organized around several female-headed, multi-generational families, or matrilines, plus males who come in from the outside, and this arrangement derives from the fact that females stay in the group in which they're born for their entire lives, whereas males stay only until puberty, and then around puberty they leave, either voluntarily or they're kicked out of their group. They then typically join all-male gangs, hang out in those gangs for anywhere from several months to over a year, and then work their way into a new troop. So every troop has plenty of adult males and adult females in there, but the females have all been there since birth, and they're all related to one another, at least within their respective families, whereas the males initially are related to no one until they start having offspring of their own. And here's one such family. Here's the mother, newborn infant. There's a grandmother back there. This is a older sister of the infant. This is a younger sister of the mother. And the other thing that you should know about rhesus monkey social organization, which I'll come back to later, is that within every group, there are multiple dominance hierarchies, differences in social status, such that every member of the highest ranking family, including infants, outranks everybody in the next highest ranking family, including adults. There's a hierarchy within each family. I won't go into the details, but basically it's that younger daughters outrank older daughters. There's a hierarchy among the males that come into a troop that's roughly related to tenure. That is, the longer a male is in a troop, the more likely it is to be high ranking, but of course, it's not tenure or length of time. What's really important is how good those males are at making friends and sustaining alliances, not only with other males, but especially with high-ranking females who carry the real clout in the troop. And males that are good at that stick around and move up, and males that are not so good give up, leave, and seek their fortune elsewhere. And then finally, there's a hierarchy among the infants who grow up, who are born into the troop every year, that roughly is in the same, reflects the position of their mother. So high-ranking mothers have high-ranking infants, and low-ranking mothers have low-ranking infants, with a few exceptions, but that's the general rule. Just a little bit of background on how these monkeys grow up. By the way, they grow up 4 times faster than we do, which is an enormous advantage for those of us who are interested in studying the long-term effects of early experience, or lifespan development, or the transmission of characteristics from one generation to the next. We can see a generation in 4 or 5 years instead of having to wait 15 or 20 for the human equivalent. So when I give you an age in rhesus monkey months or years, just multiply by 4 and you'll get the rough human equivalent. At any rate, rhesus monkeys in the wild and at our Poolesville facility spend virtually all of their first month of life in intimate physical contact with their mother, and during this time, a strong and enduring attachment bond is established between mother and infant. And this bond is functionally equivalent to the kind of bond that human infants routinely form with their caregivers in virtually every culture in the world. As these monkeys grow up, once that attachment bond has been established, these monkeys will then begin to venture away from their mother to start exploring their world, and they'll do it in a very stereotypic fashion. They'll use their mother as a secure base. So an infant will move away from his mother for a few seconds or minutes, then running back to the mother for a period of contact, will then go and explore some more, come running back to their mother for a period of contact. So it's almost as if the infant is attached to its mother by a long rubber band. And as the weeks and months go by, these exploratory forays become more frequent, involve greater distances away from the mother and more time away from the mother, so that by 6 months of age, these youngsters are spending only about 20% of their waking hours in actual physical contact with their mother, as opposed to the 100% that they were spending in that first month. Yet the mother's presence in the immediate vicinity is absolutely crucial to sustain this early exploration, because in the absence of access to your attachment object, you'll get very emotionally upset, and any exploration will cease. So even though these youngsters are not actually physically contacting their mother that much by the time they're 6 months of age, their mother's presence is a crucial part of their social world. Who are these infants interacting with? Well, they're interacting with peers. And in the wild, this is not by accident. In the wild, rhesus monkeys are seasonal breeders. They copulate year-round, but the actual conceptions only occur within a 2 to 3 month period every year. So that means 80 or 90% of a troop's infants are born within a 2 to 3 month period each year. And what it guarantees is that any infant born during that period will have plenty of other youngsters the same age and roughly the same physical and cognitive capabilities as themselves, and that's who these youngsters prefer to hang out with. Throughout their childhood years, the rest of the first year of life, the second year of life, and the third year of life, these monkeys will spend several hours every day in active social play with their peers. So that's what happens in the wild and in our field station. But not all monkeys at Poolesville grow up that way. Some of them are reared by their biological mothers in smaller social groups. Others are cross-fostered to unrelated females, so we can start separating out genetic and environmental components of their early background. And others are reared without mothers. These are infants who grow up using a paradigm that my mentor, Harry Harlow, many years ago initiated. It's called peer rearing or together rearing. Here, infants are separated from their mothers at birth before they have any opportunity to form any kind of a social attachment to the mother. They are hand-reared in a neonatal nursery by human caregivers who spend probably 10 to 12 hours actively interacting with them during the first month of life. And then they're moved into small groups of peers, like reared, 4 or 5 in each group, where they stay with their peers 24 hours a day until they're about 6 or 7 months of age, at which point they're moved into larger social groups that also contain their mother-reared counterparts who have been weaned at that point, as happens in the wild at 7 months of age. In the wild, mothers wean their infants to get ready for next year's infant. So from 7 months of age onward, mother-reared and peer-reared monkeys are living in exactly the same environments. So any difference that we see in their behavior, biology, or gene expression can be traced back to that initial 6 months of life. When monkeys are reared in this fashion, they become hyper-attached to one another. Very, very strong bonds among the infants, but these bonds, while very strong, are actually somewhat dysfunctional because they spend all of their time clinging to one another rather than going out and exploring their environment. And when they get to the age where they should be starting to interact, to play with peers, in fact, they play a little bit, but the level of the play and the intensity and the complexity of the play never reaches the level of their mother-reared counterparts. And it's not surprising because they have to be serving as attachment objects and playmates at the same time, and they don't do a very good job of either. So that even though these peer-reared monkeys are with potential playmates 24/7, they actually are play deprived as they're growing up. And this has some consequences. We now know that these monkeys, as a consequence of their early rearing, tend to be excessively fearful. And in that respect, they resemble the 20% of the population in the wild who are excessively fearful and anxiety and depression prone. They also show heightened neuroendocrine response, stress response to minor changes in their environment relative to their mother-reared counterparts. They also, at the same time as they get older, become more impulsively aggressive. And in that respect, they resemble the 5 to 10% of the population in the wild that seem to be excessively aggressive. And like that 5 to 10% in the wild, they have problems with their neurotransmitter metabolism. In particular, they tend to have poor serotonin metabolism relative to their mother-reared counterparts. We've done neuroimaging studies with these monkeys, and the brains are different than those of their mother-reared counterpart. Here's PET scans of a composite of mother-reared versus peer-reared, top view of mother-reared versus peer-reared, side view of mother-reared versus peer-reared with respect to serotonin transporter binding, and similarly for cerebral blood flow, mother versus peer, mother versus peer, mother versus peer. Simple inspection suggests that the peer-reared monkeys' brains are not lighting up as much as their mother-reared counterparts, and when you look at the actual data, indeed, throughout the brain, this is the case. Whether you're looking at the raphe, the thalamus, the striatum, the temporal or the frontal regions of the brain, peer-reared monkeys have significantly less serotonin binding potential than their mother-reared counterparts. So peer rearing has the effect has consequences for behavior and emotional regulation, more fear, more aggression. It has consequences for their neurotransmitter metabolism, metabolism, lower levels of serotonin, for their neuroendocrine response, higher levels of the stress hormone cortisol, and even differences in brain structure and function. And in that respect, and where the tie to the human literature comes in, is that they look very much like offspring of those cases in the wild and in other laboratories where there is maternal neglect and abuse occurring in a natural setting. About 10% of the monkeys, females in the wild, neglect their offspring, and their offspring look very much like pure-reared monkeys with respect to their behavior: more fear, more aggression, higher cortisol, lower levels of the serotonin metabolite. So pure rearing is somewhat of a proxy for maltreatment maternal maltreatment that occurs naturally. And again, it's about 10% of the population in the wild. Recently, we have been looking at patterns of gene expression and gene methylation, as David referred to. We have two sets of collaborators. One of them is our individuals, Steve Cole at UCLA, a brilliant molecular biologist, and James Heckman at the University of Chicago, a Nobel Prize-winning economist. And what Steve does is looks at patterns across the genome of gene expression in a certain type of white blood cells called leukocytes. And let me walk you through this graph. These are data from his analyses of genome-wide expression in leukocytes between mother-reared and pure-reared monkeys. Every vertical line is a gene. Every horizontal line is a monkey. So mother-reared, 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 pure-reared, 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 pure-reared. If you see green, that gene is underexpressed. If you see red, that gene is overexpressed. And what you see is there's almost no overlap in all of these genes, with one exception. That's MR55, whose genetic profile looks very much like that of a pure-reared monkey, but it's mother-reared. It turns out that MR55 had a mother who was neglectful and maltreated that particular infant. So again, you see that same, in this one case, that same parallel between maltreatment and pure rearing. Now, these are a lot of genes. I don't know what they all do, but Steve Cole does. He's been able to point out that those genes that are overexpressed in the pure-reared monkeys have to do with inflammation, cell growth and differentiation, and transcription control. The genes that are underexpressed in pure-reared monkeys have to do with immunoglobulin production, interferon, antiviral response, and antibacterial response. And you may say, well, that's nice, but what does it mean? Well, Steve Cole knows, because his day job is not to look at white blood cells in monkeys, but to look at white blood cells in humans. And some years ago, he published a paper with a brilliant social psychologist from the University of Chicago, John Cacioppo, looking at adult humans who characterize themselves as either being socially isolated, lonely, or well socially integrated. And if you look at those patterns in the lonely individuals that are overexpressed, it's inflammation, cell growth, and transcription control. The ones that are underexpressed, immunoglobulin production, type 1 interferon, and antibacterial response, exactly the same as you see in the pure-reared monkeys. Now, I'm not about to stand up here and tell you that pure-reared monkeys, adult humans who consider themselves socially isolated, are exactly the same thing, but at least the pattern in their gene expression in white blood cells is strikingly similar. The other collaboration we're doing is with Moshe Ziff, whose picture you saw earlier, looking at methylation. Methylation is I won't go into the details, but it's a process by which the addition of a methyl group to a particular part of a gene will essentially shut that gene off, as in this case. And Moshe does genome-wide methylation studies of leukocytes, another type of white blood, lymphocytes, excuse me, another type of white blood cells. And here's a young Moshe Ziff, younger than what the slug that David had. And he looks at genome-wide patterns of methylation as a function of differences as a function of early experience. And in the study we published a couple of years ago with his group, we found that fully 1/5 of the entire rhesus monkey genome is differentially methylated as a function of what happened in that first 6 months of life, whether the infants grew up in a nursery or grew up with their biological mothers. And more recently, and those patterns last up until adulthood in the absence of any kind of intervention. Well, since that initial study, we've done some intervention studies where we've tried to improve the environment of these young monkeys as they make the transition from their early rearing to being reared, living in mixed groups. And one of the procedures we've used involves putting within the newly formed groups what we call foster grandparents. That is, old adult females who are postmenopausal but have a history of being very nurturant towards the offspring that they reared themselves, and old adult males who also have a history of positive interactions with the infants. And what we find is that these old females, these foster grandmothers, are there to comfort infants, youngsters who are having trouble with this transition. And because aggression tends to increase in these newly formed groups as they get older, the males are there to keep the peace, to break up fights. And here's one of our foster grandparents. This is a male. This is Doogie. Doogie's going to keep the peace. Nobody's going to mess with Doogie. When we do that, what we find is remarkable. We find that the differences in methylation patterns that are enormous after 1 month of age. So over 5,000 genes are differentially methylated depending on whether you grow up in a nursery or you grow up with your mother, already by 1 month of age. Our intervention happens around here, and by 2 years of age, these differences have greatly diminished. In the males, they're not 5,000 anymore, they're 2,500. That's half of what it was before. So what this means, and we now know the specific genes where this is occurring, is that those pure-reared monkeys' pattern of gene expression are more closely resembling those of their mother-reared counterparts after this intervention. And if you look at females, it's not 5,000 anymore, it's not 2,500, it's about 750. Now that's still a lot of genes, but it's 1/6 of what it was at 1 month of age. So again, this intervention seems to be normalizing not only the behavior, which we can measure, and the stress hormonal response, which we can measure, but also in the very pattern of gene expression and gene methylation. So these patterns, that's what's causing it all, these patterns appear to be reversible by environmental intervention, social intervention. So peer rearing is one form of early life adversity that has major consequences behaviorally, biologically, and in terms of gene expression and methylation. But it seems to be, at least in part, reversible by various intervention. Let's look at another type of early adversity. I mentioned at the outset that these monkeys in the wild live in social groups where there is a very distinct social hierarchies. You have high-ranking individuals and you have low-ranking individuals. And you have high-ranking families and low-ranking families, and we know that infants who are born into high-ranking families tend to be high-ranking, at least initially, and infants that are born into low-ranking families tend to be low-ranking, at least initially. And we know that this characteristic is transmitted probably epigenetically, certainly not purely genetically, from one generation to the next. So that high-ranking females, as I said, tend to produce offspring, female offspring, who themselves will grow up to be high-ranking. Low-ranking females tend to produce offspring who themselves will grow up to be low-ranking. How does this happen? Well, we now know that if you look at the genomes, methylation-wise, across high-ranking and low-ranking individuals, the high-ranking individuals, females, have a very different pattern of genome-wide methylation than the genome-wide methylation pattern of the low-ranking females. And when you look at their offspring, you see the same pattern, at least very early in life. So recently, in collaboration with Moshe Ziff, we have started looking at placentas. That is the thing that fetuses develop that helps regulate their early development. And it's possible, If you collect placentas, you can also look at genome-wide methylation patterns in the placenta, and that's what Moshe Zif's group has done. Let me step aside for a second and say that maternal rank does not necessarily mean incompetent or abusive or neglectful females. I'll show you one example. Here is a very low-ranking mother with a 6-week-old infant who's watching the infant want to get away from her to go and check out a fight. The mother knows that if she gets into that, if she lets her infant do that and it gets in trouble, she doesn't have the social clout to go rescue it. So what does she do? She grabs the infant, she picks up a flower, she distracts the infant, and then once the fight is over with, she then allows the infant to go and explore in an area that's now safe. Let me play that again. There's the infant looking at the fight. Mother grabs the infant, grabs the flower, distracts the infant, does face-to-face. You look in the right-hand corner and the monkeys in the fight are now disbanding. Face-to-face interaction. Now the coast is clear. Now the mother is able to let her infant go explore where it wanted to. She even gives it a little push, and then she follows it just to make sure. That's a really good example of a very cool mother, even though she's very low ranking. So maternal competence and social rank are somewhat independent. We know that some of the highest ranking monkeys in the wild, in high ranking families, routinely have neglect and abuse. So again, mothering characteristics and social rank are somewhat independent of one another. The interesting thing with the dominance ranking is that if you are a high-ranking female and you do have good maternal characteristics, you have the time and the freedom to spend more time interacting with your infant than would a low-ranking mother. So here's a high-ranking mother. And now, without any worry about other monkeys interrupting, she can interact extensively with her infant and make it much, we now know, put it in shape to become better at socializing with other individuals besides her. So dominant status can be important, and we now know it's probably as important in terms of the number of genes that are affected, as is the case with differential early experience. And so we can, if you want to look at social adversity beyond maltreatment, just because you're high-ranking, low-ranking means that you're going to have restriction on your social activities and your freedom to go and explore the environment. You're going to have biological consequences in terms of higher levels of the stress hormone, among other things, and you're going to have health consequences, higher injury rates, immune system vulnerability, excessive inflammation, high incidence of gastrointestinal disorders. So being low-ranking is a risk factor for sure, and it happens in every monkey social group because not everybody could be high-ranking. One of the things we're looking at is how is this transmitted? We know it is transmitted from one generation to the next. High-ranking females have high-ranking female offspring. Low-ranking females have low-ranking female offspring. Moshe Zif 2 years ago told me to start collecting placentas. I didn't know why. Now I do. Because if you look at the placentas, there are high-ranking placentas and low-ranking placentas. So here's another one of these heat plots. If you see, these are high-ranking individuals, mothers. These are high-ranking placentas, sorry. These are low-ranking placentas. If you see red, genes in the placenta are overexpressed. If you see blue, genes in the placenta are underexpressed. There's almost no overlap, even though the N is very small. So the placentas of these females and their offspring are as distinctive with respect to social rank as themselves. What this means or suggests is that here is a biological mechanism by which something social status can be transmitted from one generation to the next. Now this has some pretty interesting implications. Among other things, the pattern that you see in these placentas are very reminiscent of what you see in individuals who were mother-reared or peer-reared. It's the same set of early adversity genes that are being differentially affected. And we now know that we can do something about this, at least in our monkeys. It's possible to change dominant status relatively easily, independent of early experience, so it's not that difficult to make high-ranking females lower ranking or make lower-ranking females higher ranking, and we can see whether their pattern of methylation changes, whether their placenta's pattern of methylation changes, and what are the effects on their offspring. And furthermore, we can change the dominant status of their offspring. If you do that, does their pattern of methylation change? In other words, is the social environment really affecting the way these genes are being expressed and methylated? So we have a number of things on our agenda for the near future. We certainly, as I mentioned, can experimentally manipulate social dominant status. We can also look at, I think, something that some of the demographers in human research are starting to look carefully at, and that's the steepness of the gradient, the difference between high-ranking and low-ranking individuals. In some of our monkey groups, the higher-ranking females are very, very dominant relative to their lower-ranking counterparts. In other social groups, the difference is much smaller. Are there health consequences of those differences in the steepness of the social gradient? We can look at that as it occurs in nature or in our groups. And finally, if we manipulate that and either increase the slope or decrease the slope, are there health consequences, as well as the behavioral and biological ones that we seem to expect. So in all of this work, we are guided by the human literature, asking questions that are difficult to answer directly in humans, but I think we can do in monkeys. These are some of the people that have been working on this problem over the years, and this is my lab as it is today, and thank you for being patient and hearing all these bumps in the vocal tract.